Hi everyone, today I will be showing you how to extract DNA from a strawberry, and I will be explaining why this process occurs. The things you will need to complete this experiment are 1 tablespoon of dish soap, half a cup of ice-cold isopropyl alcohol, a large and a small clear container, a Ziploc bag, some strawberries, half a teaspoon of salt, a third cup of water, some tweezers, and a strainer. First, pour the third cup of water into the larger container. Then put in a tablespoon of dish soap. Also put a half a teaspoon of salt in. Stir the solution around. Next, place some strawberries into the Ziploc bag. Pour the solution from the large container into the Ziploc bag. Seal the bag tightly so there is no air left in the bag. Next, crush up the strawberries so there are no large chunks left in the bag. Next, place the strainer on top of the smaller container and pour the strawberry solution through the strainer into the smaller container. Lastly, pour half a cup of ice-cold isopropyl alcohol into the small container. This is what the solution looks like now that we have added everything into it. Using your tweezers, skim the surface of the solution and pick out a light pink material. This is the DNA of a strawberry. But how does this work? First, strawberries were used because strawberries are octoploid and contain 8 sets of each of the 7 types of chromosomes found within strawberries. This therefore means that strawberries contain a lot of DNA. The strawberries are squished in order to break down the cell walls of the strawberry cells and release the contents of the cells. Soap is added to the strawberries to dissolve the cell membranes and nuclear membranes of the strawberry cells. Soap molecules have a similar structure to the phospholipids found in the cell membranes of strawberries, but the soap molecules contain only 1 hydrophobic tail, unlike the strawberry phospholipids which contain 2. The soap molecules form an aggregate of molecules known as micelles. When the micelles come into contact with the strawberry cell membranes, the phospholipids come apart, thus resulting in the breakdown of the cell and nuclear membrane. Salt is also added to release the DNA strands from the proteins that they are tightly coiled around. These proteins are positively charged and are displaced by the sodium ions found in the salt, which allows the DNA to become separate from the rest of the strawberry cells. Lastly, the strawberry solution is placed in ice-cold isopropyl alcohol. DNA is not soluble in isopropyl alcohol, which therefore allows the DNA to separate from the rest of the mixture and float to the top of the glass. The isopropyl alcohol is cold in order to slow down the reaction and make it last longer for us to see. I really enjoyed extracting DNA from a strawberry. I never knew that it was possible to view the DNA of an organism without using a microscope. The parts of the experiment that went well were mixing the correct solutions together and eventually extracting a lot of DNA from the strawberries. The parts of the experiment that didn't go well were measuring the correct amount of each solution, as they had to convert everything in milliliters to cups, tablespoons, and teaspoons, which isn't as accurate. In order to make this experiment better, I can use a measuring device whose units is in milliliters.